Hi, grade 10. So what I'd like to do today is to go through the basic stages of cell division and what happens, or the phases of cell division and what happens at each point. Alright, so you might remember that we said that when a cell is not dividing, that it's called interphase. Okay, and remember that the nuclear material, the genetic material in the nucleus, it's DNA, but when the cell's not dividing, it's more thread-like, all right? So we call it chromatin, thread-like chromatin, but it's made of a molecule called DNA. And here you can see that, okay? So there's the nucleolus, there's all the thread-like DNA, all right, tangled threads. I wouldn't even say chromosomes yet. It's tangled threads forming the chromatin network. In other words, lots of threads in the nucleus, which is the cell is not dividing. All right. So now the cell has to prepare for cell division, and the first stage that happens is the nucleus divides, and then the cytoplasm divides. So if you remember from the introductory video the other day, we talk about IPMAT. So interphase is when the cell is not dividing, but it also starts preparing for cell division. In other words, the DNA prepares. Then you've got the actual stages of mitosis, all right? And that's where the genetic material is divided between the two daughter cells. Then right at the end, we've got something called cytokinesis, and that's where the actual cytoplasm of the cell splits, all right? Then over here, this is karyokinesis, okay? Karyokinesis is when the DNA is divided between daughter cells, all right? So if we look here, when the cell is not dividing, the DNA is thread-like, it's called chromatin. Then if the cell is preparing for cell division, each strand of DNA coils up to form more organized chromosomes, okay? But now hang on, if this cell is dividing, each daughter cell needs to get a copy of the original DNA. So the cell that divides is called the mother or the parental cell, all right? The DNA molecules coil up to form chromosomes, and then DNA replicates. All right, and in grade 12 you learn how this happens, but basically, I'm just going to draw it at the top here. Remember, the DNA molecule is like a ladder, so there are enzymes which break that ladder apart. All right, so enzymes break the ladder apart, they break the bonds between the nitrogen bases, and then new nucleotides, and you don't have to understand all of this, all right? New nucleotides are brought in, so that ultimately you end up with two DNA molecules, all right? I'm just trying to show now, okay, that the original DNA molecule separates, all right? The nucleotides are brought in, and they join onto both sides of the original DNA molecule, so you end up with two DNA molecules. And remember, we said that, okay, wait, let me rather draw that over here. So there's one DNA molecule, there's the other DNA molecule, and they're joined by a region called the centromere. So this is one replicated chromosome. Remember, you count chromosome number by the number of centromeres, all right, not the number of DNA molecules. So when the chromatin thread coils up, that is one chromosome. There's the centromere. Then DNA replication happens, and you end up with two DNA molecules. So both of those coil up, and they both consist of half old and half new nucleotides. That's grade 12 work. And they held together at a region called the centromere. So that is one replicated chromosome, all right? So the DNA has been copied. So if we look here, this happens at the end of interphase and in prophase. Now, cells don't decide, okay, I'm in interphase, now I need to divide, now it's prophase. Okay, stop prophase, hang on, we're going to move into metaphase, then we're going to move into anaphase and telophase. No, it's not like that. All right, we like to name things. We like to categorize everything. If you go into a shop, you know, the veggies are one side, the meat is one side, the frozen food, the dairy, the cereals, etc., etc. All right, so prophase is when the cell, P for prepare, it prepares for cell division. Okay, you can see here the chromosomes are becoming more visible, the DNA molecules are coiling up, the threads are coiling up and they're becoming shorter and thicker. Now, we don't really have to distinguish between early and late prophase, all right? I'm not going to worry if you get the late or the early right, but here you can see that the DNA molecules are now replicated and they each consist of 2 DNA molecules joined by a region called the centromere. All right, so there are 2 chromatids. Okay, each of those DNA molecules are known as a chromatid. All right, now I want to go back a bit. In the beginning here, all right, there would have been, in this particular cell, there would have been 4 strands of DNA, the chromatin, and the reason why I'm saying that is because 4 strands, long strands of DNA coiled up to form 4 chromosomes. 1, 2, 3, 4. Okay, now how many chromosomes do human cells have? Well, there would have been 40 strands of DNA coiling up to form, 46 or 23 pairs, so coiling up to form 46 chromosomes, etc. But that's a lot to draw. Now, when you answer questions about this, you've got to look at the picture, or are they talking about human cells? So if they've asked you to look at the picture and there are 4 chromosomes, then that's what you talk about. If there are 8 chromosomes, then that's what you talk about. If they're talking about human cells and we know that there are 46 chromosomes, then that's what we refer to in the questions. All right, so here the DNA molecules have coiled up to form chromosomes. Here, DNA replication has taken place. So now each chromosome, that's still one chromosome, it's a replicated chromosome, consists of 2 chromatids joined by the centromere. So you count the number of centromeres. The other thing that you can see here, all right, go back to the beginning. Okay, these are the centrioles. Now you should remember from the cell is that the centrioles are cylindrical bodies which lie at right angles to each other in a region known as the centrosome. So those are the 2 centrioles in a patch of cytoplasm, and we call that the centrosome. You can see here that what actually first happens is that they replicate. So you end up with 2 pairs of centrioles which start moving apart and little fibers start developing between them. They're called spindle fibers. You can see here in late prophase that those centrioles are now moving further apart. You can also see that the nuclear membrane is starting to break down. So here it's even more broken down, and even the nucleolus breaks down. Now the nucleolus is just a region where RNA is stored. Now, why does the nuclear membrane break down? Because the chromosomes have to be able to move. It's a membrane. It's proteins, it's lipids, it's cholesterol. So the molecules break down, they move apart, they become part of the cytoplasm, allowing for these chromosomes to move around. All right, so eventually the centrioles are going to reach the poles of the cell. All right, so only animal cells have centrioles. These are not found in plant cells. Okay, and what happens is these spindle fibers develop between them, like little elastic bands that develop across the cell, all right, and they are going to attach to the centromeres of each replicated chromosome. All right, so this was prophase. Then when the chromosomes line up along the middle or the equator of the cell, okay, we call it metaphase. So there are 4 replicated chromosomes lining up along the middle of the cell. If we're talking about a human cell, think of the Earth. The equator is across the Earth. There would be 46 chromosomes lined up along the middle of the cell. And when you watch the video on mitosis that I upload, real life mitosis, you will, it doesn't happen so prettily as this. This is a lovely little diagram. It's been really nicely drawn, but in real life it's not going to be as neat as this. Okay, so the chromosomes are lined up along the middle of the cell, the equator or the equatorial plane of the cell, and the spindle fibers attached to the centromeres. Okay, they line up along an imaginary line called the equator, and the chromosomes attached to the spindle fibers by their centromeres. Remember, the centromeres are where the chromosomes are being held together, okay, by my palms over there. Right, now, so we go this way now. Okay, so it's interphase, prophase for prepare, metaphase for chromosomes lining up along the middle of the cell, the equator. Then it's anaphase for moving apart. So what happens now? Okay, there was a chromosome, there's the other DNA molecule, chromatids, and they're held together by a region known as the centromere. Okay, so that's a replicated chromosome. So what's going to happen? Sorry, that's my phone. I just want to put it off quickly. Okay, so there's the replicated chromosome, there is the centromere. So what happens in anaphase is the centromere divides longitudinally. Okay, then each chromatid or DNA molecule is going to be pulled apart. So in anaphase, the spindle fibers contract and they pull these chromatids apart. But now you can actually call them daughter chromosomes because they're going to become part of the daughter cells. So if you look here, there were 4 chromosomes at the beginning. They were replicated. So 4 chromatids or 4 daughter chromosomes are pulled to each pole of the cell where the centrioles are in animal cells. The spindle fibers contract to pull them towards the poles. Okay, so that's anaphase. Spindle fibers contract and pull the chromatids, or we can call them the daughter chromosomes, towards the opposite sides of the cell called the poles. Okay, then over here. T is for telophase. In other words, you end up with 2 nuclei. Okay, so you can see here these daughter chromosomes collected one side, these 4 collected the other side, and the nuclear membrane reforms around them. The nucleolus reforms in each daughter nucleus. Alright, so now you've got 2 nuclei, the end of karyokinesis. The genetic material, the DNA, is being divided between the 2 cells. So like I said earlier, the cell doesn't say, okay, metaphase, right, stop, anaphase, start, etc. So you can even see here that the cytoplasm is start to divide between the cells. So that's called cytokinesis. Invagination or constriction of the cytoplasm occurs. It's almost like taking a blob of Prestic and splitting it into 2. Okay, so cytokinesis is when the cytoplasm splits and you end up with 2 daughter cells. So let's see here. Early telophase, the chromatids or daughter chromosomes have reached the poles, nuclear membranes form, and you can see, the spindle fibers are starting to break down, and each cell will have its own set of centrioles. Okay, over here, late telophase, you can see that the DNA and the chromosomes have untangled. So now we've got chromatin inside, 4 strands for this particular cell, 4 strands of DNA or chromatin in each cell, and cytokinesis is completing, whereby the cytoplasm splits between the 2 cells. All right, so the chromatids, you can call them daughter chromosomes. I even started calling them daughter chromosomes over here. All right, they unwind, they become long threads of DNA, long thread-like DNA again, and you're going to end up with 2 daughter cells, and they're identical to the original cell because there are 1, 2, 3, 4 chromosomes here, and there were 4 chromosomes in the original cell, and the reason why that could happen is because of DNA replication. But hang on, what about plant cells? Now, plant cells are surrounded by a cell wall. So everything happens the same except for cytokinesis. It's a bit of a problem if it's a plant cell because of the cell wall, the rigid cell wall around the cell. So at the end here, end of telophase, there would have been 2 nuclei, all right, with the chromosomes and the nucleolus inside each nucleus. And what happens is that cellulose is deposited in the center, in the middle of the cell, and it grows sideways. That plate, it's called a cell plate of cellulose, grows towards the edges of the cell. So you ultimately end up with two daughter plant cells, each with their own cellulose cell wall and their own nucleus. So that's the difference in plant cells, is that a cell plate of cellulose separates the cytoplasm into two. I sometimes think of, the magicians, where they pretend that they're cutting, a lady lying in a box, and they pretend that they're cutting the lady in two, but actually there's two ladies. They're just all, folded up really tightly. Okay, so that's how mitosis occurs in animal cells, and you need to understand at the end here, the difference in cytokinesis of plant cells.